Deepak Gupta, and I'm pleased to welcome you to our President's Council of Fall Symposium. I hope you guys enjoyed the wine outside and the CRISPR-modified hors d'oeuvres. Not really, not yet, but you might hear about how that may be the future. We'll load them up with goodies for you. I see a lot of new faces in the audience, which is wonderful. And so I thought I would just take a moment before we launch into our really wonderful program that we have for you today to tell those of you who are new a little bit about Gladstone so that you know what we're all about. So the Gladstone's mission is simple. It's science overcoming disease. We do biomedical research with the express purpose of understanding how and why disease occurs so that we can fix that. And our size, we're about 500 employees in the building, so there are a lot of human diseases. We can't cover everything. But instead, what we've chosen to do over the years is to focus our efforts on a few major diseases of mankind and build relatively small teams, but with critical mass, to go tackle that and make a difference. And in each of the areas where we've focused, I think we're arguably among the top 3 to 5 places in the world contributing to understanding those areas. And those are cardiovascular disease, neurodegenerative diseases like Alzheimer's, Parkinson's, and viral and immunologic disorders. And we've recognized more and more in the last several years that as science has reached an inflection point where we're finally beginning to understand human disease at a deeper level, for many of the things you'll hear about today, technology is driving this forward more than anything. And you're going to hear about one of those technologies today, but there are a number of them. And so we have taken a purposeful effort to bring in cutting-edge technology, data science, to analyze the big data that comes out of those technologies and put those to work to understand human disease. And we sit in a place geographically where we are able to take advantage of a wonderful relationship we have with UCSF across the street. And scientifically, it's extremely valuable. But we are independent in that we are financially and administratively separate, which allows us to be scientifically seamless with the larger entity, but control our destiny. And that's been extremely valuable in many ways, particularly as we go to take our discoveries and be a bit more entrepreneurial than one might be otherwise, and actually make sure that those things get out when they've got legs to try to help people. So that's the approach we've taken. And as we do that, we've tried to build a culture where we are taking risks. We're going for the big problems and trying to figure out what if we don't, nobody else is going to. And to do that, it really takes some risk capital. And we've been able to do that by a healthy endowment, by the efforts of the President's Council that also makes this event possible twice a year. So many of you in the room are the ones who are actually been enabling us to be at the forefront and take those risks and really make a difference. So with that, let me just take a moment to frame the topic today, which is timely. You hear about it in the news. It's all over the place, and it's changing the world. So hacking the genome. I'm sure all of you knew that back in 2001 that we had sequenced the first human genome. And there was a lot of enthusiasm and hope that now we're going to be able to figure out human disease and fix this and fix that. And then there was a pause, right? There was a few reasons. We could read the genome, and in the subsequent years, it became cheaper and cheaper to read the genome. And now we can figure out any one of your genomes in the audience for less than $1,000. So you can read it. But if you could just read it, then what are you going to do about it? You have to understand it, and you have to be able to manipulate it. And so I'd argue that there have been, in addition to being able to sequence genomes, there have been 2 major breakthroughs in the last decade or so that have really enabled us to understand human disease and human genetic variation like we could never before. One of those was the discovery of induced pluripotent stem cells, or iPS cells, where we can take blood cells or skin cells from an adult, any one of you, and convert that into a stem cell in a dish. Now, it's your stem cell that we can make into any one of your cell types. So that, for the first time, allowed us to take human cells in a dish and figure out why the genome, if it is altered to cause a disease, was causing a problem. We could never do that before in a human cell. That discovery won the Nobel Prize in 2012 and was made by Shinya Yamanaka, one of our faculty here. But there was a limitation. You could read the genome, you could understand it, but if you couldn't do something about it, then you're stuck. And so what really opened up the field was Jennifer Doudna's discovery that you'll hear about today, which is using CRISPR technology to actually edit or rewrite the genome. And so now you can read it, you can understand it, and you can manipulate it and rewrite it. And that technology together is so powerful. And we're delighted that this summer, Jennifer Doudna, who has been at UC Berkeley and done the amazing work she's done in discovering CRISPR technology, chose to start a lab here at Gladstone, and she'll tell you more about that in a moment. But it's going to allow us throughout the institution and the campus to more closely marry the ability to read, understand, and rewrite the genetic code. So you're going to hear today from Jennifer, who I'll introduce now, and then we've got two other panelists that you'll hear from, Rachel Haurwitz and Bruce Conklin. And I'll introduce them in a moment when we're all on stage. But Jennifer, as I mentioned, even discovered CRISPR technology, but even before that, had an illustrious career really understanding the genome and parts of it that other people hadn't really studied so much, particularly the RNA of the genome. She's received numerous prizes. She's a member of the National Academy of Sciences, a member of the Howard Hughes Medical Institute. She was one of Time Magazine's 100 most influential people in 2015 for her discovery. She's a recipient of the Breakthrough Prize, which we'll celebrate here in the Bay Area just next week. And we're delighted to have her as our newest Gladstone investigator. Jennifer?
Well, thank you very much, Deepak, for that generous introduction, and welcome everyone to Hacking the Genome. It's a great pleasure for me to have the opportunity to talk with you a little bit about science that I've been involved with, and also to tell you a little bit about my vision for the future and why did I join the Gladstone. And so I've been a faculty member across the Bay at the University of California, Berkeley for 16 years. And before that, I was on the Yale faculty, so I've actually now been running a lab for 24 years. And during that entire time, I've focused on very fundamental questions in biology, some of them kind of obscure questions, trying to understand the functions of molecules called RNA that play a critical role in the transfer of information between the genome, the DNA of a cell, and all of the molecules like proteins that carry out most of the cellular functions. And I've always been fascinated by that that flow of genetic information and the role of RNA in that process. And I didn't at all start out with a vision of creating a technology for genome editing. That wasn't our goal at all in the lab at the time. But in fact, we were interested in understanding the fundamental way that bacteria interact with viruses in the environment. And so I'm going to just show you, I want to just walk you very briefly, very, very briefly through our thought process and what CRISPR technology is, a little bit about how it works, and then where I see it going. And this is where my decision to join the faculty here at the Gladstone comes in. So on the next slide, I think the story of genome editing, in a way, really begins, we go back a few decades, back to the 1950s, when scientists first discovered the structure of DNA, the double helical structure that holds all of the genetic information for all of life. It's a profound, it was a profound discovery at the time, and it really helped to explain how genetic information is transferred from one generation to the next. And I think really, at that time, in the 1950s, scientists already started imagining, what if you could read that code? What if you could write that code? What if you could rewrite that code? And when I think back to when I was a graduate student in the 1980s, there were already scientists then thinking about how to manipulate DNA sequences in cells in a precise way. And over the last few decades, there have been a series of technologies for doing that have come along that have shown us two things. One is that it's very powerful if you can manipulate the DNA sequence inside of a cell. And secondly, it's not very easy to do. And so on the next slide, this is an illustration of how this technology that we now call CRISPR for editing genomes came about from a very unexpected direction. Like I said, it wasn't a technology that started off by somebody sitting in a lab and saying, I'm going to think about the best way to edit genomes. It was actually a group of scientists who were kind of dispersed around the world. Some of them were working in companies, and some of them were working in academic labs like ours, who were asking questions about the process by which bacteria fight viral infection. And this is a picture of you're looking at a section of a bacterial cell being attacked by viruses that land on the surface and inject their DNA. They inject their genetic material into the cell and take over the cell's machinery and start making more viruses. Same process that happens when we get infected by a influenza or any other type of virus. And so in my lab at Berkeley, I was studying this process. And in the course of that research, and some of it was actually done, I think the very first student working in the lab on this pathway was actually Rachel Haurwitz, who you'll hear from. We realized that bacteria had already come up with a way to find DNA sequences and cut them as part of a bacterial immune system, a way that bacteria can defend themselves against viruses by acquiring viral DNA, sticking it into the bacterial genome in a place called the CRISPR, and then using that information to find and destroy viruses that had a matching DNA sequence should they show up in the cell again. And it was through studying an enzyme, a protein called Cas9 that's part of this pathway, that is programmed by molecules of RNA that can find and destroy particular viruses. We realized that that protein and its programmable nature, it could be programmed to find and cut a desired DNA sequence and thereby trigger cells like our own to make a change in the genome, that our fundamental scientific goals became, we sort of realized that that project had a very different outcome from what we envisioned at the beginning. It's sort of what we all dream of in science when we do fundamental work, is we hope that our results will have impacts that we can't imagine at the beginning. So on the next slide, I just wanted to show you a quick video that illustrates how the CRISPR technology operates. And so here we're zooming into what we call a eukaryotic cell, like let's say it's a human cell where the DNA is inside the nucleus of the cell. And when this protein, Cas9, and its guide RNA go inside the nucleus, this protein has the ability to search through all of the DNA in the genome of the cell to find a section of the DNA that has letters matching the letters in its guide RNA, the molecule that programs the protein. When that match occurs, the protein unwinds the DNA, cuts it, and then hands it off, hands those broken ends off to repair enzymes, repair machinery in the cell that can fix the break. And in this example, fixing it by actually inserting a new piece of genetic information at the site of the break. And what's incredibly powerful here is that this technology allows scientists to read the genetic code, the DNA of a cell, looking at, say, the entire genome of a person, and program Cas9 to find and cut the DNA at a particular place where a change to that DNA sequence is desired. And amazingly, this technology works not just in, obviously works in bacteria, but it works in a wide variety of other cells as well, essentially every kind of cell and organism where it's been tested. And so that's where the Gladstone comes in. So, I was working on this system, and when we published this work with our collaborator, Emmanuel Charpentier, back in 2012, it was clear to us as we were writing that paper and sending it off to a journal that this technology was going to be transformative. And we didn't even imagine how transformative at the time, but it was clear that there were going to be so many opportunities now that there was a way easily to alter the DNA sequence in cells. And so I started thinking about how I could take my line of work and the kind of science that I've always done and partner that with people who think very hard and very specifically about how to understand and tackle human disease. And what better place to do that than the Gladstone Institutes right here in the Bay Area. And so I had met Deepak on numerous occasions, and we started talking about opportunities for me to work together with scientists here at the Gladstone and in the broader UCSF community. And I'm proud to announce that I've just opened my second lab here at the Gladstone. So I have my lab continuing at Berkeley, but I now have a team of scientists who are beginning work here at the Gladstone to partner specifically with clinicians and look for opportunities to bring the CRISPR technology to bear on human disease. And I just want to, on the next slide, is just my logo slide that just shows that we're not only involved with the Gladstone, but I'm also, very importantly, part of and leading a group that is the Innovative Genomics Institute. Bruce Conklin is part of this as well. And I see my effort here at the Gladstone as really the Innovative Genomics Institute's effort to have a physical presence here in Mission Bay and help bring fundamental science to bear on biomedical problems. And that's really what our vision is going forward, is to partner fundamental science with clinical medicine. And on the last slide, I just want to point out, I was going to show you one quick example of science that we're already doing that we think is going to be very interesting in the context of one of the major focal points of the Gladstone, which is neurological disease. We've been working on engineering the Cas9 enzyme so that it can be injected into brains and penetrate neurons for genome editing in the brain. We're doing this, this is an example of doing genome editing in a mouse brain, and you can see a slice out of the mouse brain with parts of the tissue turning red where cells have actually been edited based on a marker that we have in this mouse. And this illustrates the principle of how we imagine in the future being able to deliver gene editing molecules into the brains of patients for the purpose of correcting underlying genetic disorders that lead to neurodegeneration. And that's a big focus of the Innovative Genomics Institute right now, is to work on that challenge. And you'll hear more about that, I think, from Bruce. And then finally, on the last slide, I just want to point out that the opportunities right now with genome editing are tremendous. It's an extraordinary time in science. As Deepak mentioned in the beginning, we're seeing this technology coming together with the fact that it's less and less expensive to get whole genome sequencing done. We have opportunities with machine learning and other kinds of computational approaches to analyzing genetic data that are incredibly powerful. And we have a very interesting and very unique opportunity right here in the Bay Area to do something extraordinary that I just don't think has been done anywhere else, even though there's lots of smart people that are using genome editing for individual applications right now. And I think all of you here that are thinking about how you might want to have an impact on biomedicine going forward, you have an opportunity to work with us to help us bring this this vision to fruition, to bring together clinical teams with fundamental scientists like myself and folks that know a lot about business and about in particular, I would say, computing and opportunities to analyze big data, to do things that none of us would be able to do independently. And that's what I'm really, really excited about doing here at the Gladstone. Thanks.
Thank you, Jennifer. I'd like to invite Bruce Conklin and Rachel Haurwitz to join on the stage, and I'll introduce them, and we can have a bit of a discussion here and then have questions from all of you. So I think as Jennifer nicely pointed out, we do sit in a unique place being here in the Bay Area with the kind of culture that we have and the entrepreneurial nature of the Bay Area and the risk-taking culture. And so I think the responsibility and the onus does lie upon us to do the right things to make sure that this technology actually makes a difference for people. Bruce Conklin is one of our senior investigators here at Gladstone. He's also had an illustrious career, and in the last several years has turned his attention to really getting the CRISPR technologies to work in human iPS cells, in human cells, and so he's been a really leader in the world in making that work, which was not trivial, and is now really trying to find ways to be able to use the technology in a person and get it used directly there to fix genetic defects. So he'll tell you a bit about the approaches we're taking there. And then Rachel Haurwitz, who, as Jennifer mentioned, trained in Jennifer's lab, has gone on to start and be the CEO of Caribou Biosciences here in the Bay Area, again, reflecting the startup culture and the nature that we have here. Rachel has been named 30 in the 30 under 30 most influential people by Forbes magazine, and then 40 under 40 by Fortune. And I'll stop there because I think that's as far as she's gotten so far. But she's really she'll tell us about how one can really take this technology in the private sector and then really the opportunities and challenges of commercializing that. So I think we'll lead off with Bruce, and you can share with the group what the approach you're taking.
Sure. This is a terrific setting. It's delightful to be here with Jennifer. We've been interacting actually since just after the discovery of CRISPR. My lab sits at the intersection between iPS cells and this CRISPR technology, and Deepak already introduced both of those technologies. But I think that the special thing that we have here is that because Shinya Yamanaka actually did his postdoctoral fellowship here, we really knew it right from the beginning. And so we had the good fortune of being some of the first to make iPS cells from patients and begin to model them. And then we were also some of the first to see the challenges that were involved. And as Deepak mentioned, the big problem here is that iPS cells from different people are different. And so, for instance, how many of you have a sibling? Raise your hand here. How many of you think that sibling is a little different than you? Keep your hand up. Okay, so we were using sibling controls for each other, iPS cells from siblings, and saying that those were controls, and that was really unfortunate, but it's all we could do because we actually couldn't genetically manipulate the cells. With CRISPR, now we could actually take those cells and actually take a cell from any one of you and then now make a single point mutation and then see in a dish how they behave differently. And we know it's due to that change and not to that difference between you and your sibling. And so we were modeling the disease, and that's when I first met Jennifer, because we were using CRISPR and genome engineering in iPS cells, and she wanted to actually move towards human disease. And I actually was, and many people wanted to take CRISPR not just to decode disease, but actually to potentially cure disease and cure a genetic disease. But I was a skeptic early on. I actually wrote an editorial or a commentary in Nature Methods saying that we're moving too fast, that we actually, that here are the criteria that we should meet before we could proceed to therapeutic editing. And then remarkably, 2 years after I wrote that commentary, I actually could see that one by one, these criteria were being met. And so I had to then essentially set upon my agenda that I'd written for myself. And I started to examine the different diseases, genetic diseases, that we potentially could use as a therapeutic now that we had met my own criteria. And we wanted 3 things now that we were to choose among these 7,000 genetic diseases. And we realized that many of these diseases we couldn't access using a traditional medical model, that it wasn't one pill for every single person that's going to work, one scalpel for every type of surgery. And actually, when you think about it, the surgical model and genome surgery is perhaps a better analogy, because a surgeon will easily use a different cut or a different scalpel for every single person. And using this sort of a surgical model of approach to this problem, we then chose 3 criteria that we really needed. One was that the disease had to be medically compelling. In other words, it had to be a win-win for the patient who's entering this a trial which is risky. And so it had to be a disease which you couldn't actually potentially, it was worth the risk for the patient. So it had to be a serious disease. The second is you had to know exactly what the target was, and you had to be able to get there. So in other words, we had to choose a disease where we knew we could actually reach that tissue, and we knew exactly what the gene is, because you can't decide afterwards, oh, maybe I edited the wrong thing. And then third is that you have to be able to measure it. And measurement is really important, because there's lots of disease which waxes and wanes, and you can't actually, as many of you are engineers, you don't want to go into a noisy system if you want to make a change. You have to have something which is rock solid. And so the diseases that we're working on are a retinal disease. It's called best one, and it's not called best one because it is the best one, but it's actually called best one because Dr. Best actually described it over 100 years ago, and it's a slowly moving genetic disorder, and in the black part of your eye, which is actually the part which we can grow and we can do the testing on and we can do delivery to. And the other one is motor neuron disease, and we're studying a motor neuron disease, and these are amazing neurons that go from your back all the way down to the tip of your toes, and we can measure their function, so we knew we could measure it. And many of you know a motor neuron disease called ALS or Lou Gehrig's disease, but Lou Gehrig's disease has a reputation for waxing and waning and moving in an uncontrollable fashion. So we chose a motor neuron disease called CMT, and CMT is a disease which moves very, very slowly, and every year it gets a little worse. And people end up in wheelchairs, unfortunately, but it's very predictable, and we know exactly what the gene is and how to get at it, and we can measure the response. And so those are the diseases that we're working. We have now 20 patients that are enrolled in these studies, and these are really in collaboration with the IGI, and we have them fully sequenced, and we're growing iPS cells, beginning to make edits and seeing effects in those cells as a result of those edits. And if those are successful, we hope to then proceed to trials in people. But right now, all the experiments are in tissue culture. So with that, I'd like to turn this over to Rachel, who's going to talk about her work.
Thanks, Bruce. Good afternoon, everyone. As Deepak mentioned, I'm the president and CEO of Caribou Bio, which is a company that Jennifer and I co-founded along with a few other scientists a little more than 6 years ago. And we are developing multiple different versions and flavors of CRISPR technology. And we're using that technology at Caribou in order to advance a variety of cellular therapies in 2 different areas, and I'll tell you a little bit about our work. The first is immunotherapy, which is the ability to actually deploy the immune system to fight cancer. And our approach to that is to use gene editing to reprogram T cells, which are specialized immune cells, in order to teach them how to find and fight cancer. Now, there are first generations of these products that are actually already commercially available. Last year, 2 products were approved, which is incredibly exciting. They are the first ever cell therapies of their kind approved here in the United States. But they have some drawbacks. They are what's called patient-specific, which means a clinician would actually take the patient's own immune cells, take them into the laboratory, provide them with a piece of DNA that then teaches that cell how to find the cancer and put it back into the patient. Now, as you might imagine, these people are rather ill by the time they have the opportunity to receive this therapy. And in fact, it's not a first-line therapy, meaning they have to have failed multiple other treatments before they're eligible for this. So their T cells are not exactly in great shape at this point. And there are a number of other challenges associated with actually manufacturing, making this therapy. And so we're very excited that with gene editing, we can circumvent this and actually take healthy cells from a healthy donor and use gene editing in order to make sure that we don't cause graft versus host disease, which is, of course, the risk when you take a cell from one person and put it into another person, and then make other defined changes to these cells to make them as good a product as possible. This is very early work. We're not in the clinic yet, but we're very excited about the progress so far. And the second main area that we're focused is what I like to call bugs as drugs, which is actually using CRISPR gene editing technology to change the genetics of the microbiome. The microbiome are the bacteria that live in and on us. They have tremendous impact on our health and on disease biology. And so we're looking at a variety of ways of actually rewriting the genetics, not of our own cells, but of these bacterial cells in order to treat a variety of different human diseases. But as Deepak alluded to in his introduction, humans are not the only organisms that are benefiting from gene editing right now. And part of Caribou's business model is that we work with a number of other companies to help enable their use of CRISPR technology in product development. And so we're involved in collaborations as broad as plant breeding to try to develop crops that are drought-tolerant, disease-resistant, or other advantageous traits, as well as animal health and well-being issues. In fact, we're involved in a collaboration, for example, with a livestock company, helping them use CRISPR technology to breed pigs and cattle that are resistant to a number of diseases for which there are no treatments today. So we're really looking at sort of the tip of the iceberg in terms of where CRISPR technology will take not only the human health field, but really any field that has bio-based products or bio-based technologies. But I do want to highlight what I see as one of the challenges of this initial commercial foray into the CRISPR gene editing world. Caribou is one of many companies who are diving all the way in and looking at a variety of applications. But as any small company approaches this field, we have venture capitalists who care very much about taking a risk-mitigated approach to developing new products and especially new therapeutic products. And so though there are a large number of companies, small and now increasingly large, moving into this space, many of us are all developing the exact same therapies in competition with one another. Because though Bruce gave us a really important statistic, there are about 7,000 different genetic diseases that are caused by only a single gene mutation and therefore potentially addressable by CRISPR technology. There's a very, very short list of diseases that we understand extremely well and where the disease-causing mutation is found in cell types that we can easily edit. And I know that's part of the conversation we'll get into a little bit more tonight. But I think it therefore highlights the importance and the opportunity at a place like here, like the Gladstone, to tackle diseases outside of where venture-backed companies can or can't take a risk. So I think it really highlights the importance of the work that my colleagues up here are doing and that many other folks here at the Institute are doing.
Great. Thank you so much, Rachel. So what I hope you can appreciate is the panel we've put together also reflects what I mentioned earlier, is engaging in deep collaboration and engaging with the private sector to really make things happen and to take advantage of the Bay Area with the wonderful venture capital efforts we have here and the philanthropic efforts. It takes all of that to do the things we need to do. So, you've heard about some of the great opportunities, but I'm sure you've also read about some of the challenges in the field, which are many, but I think are front and center, and the field's trying to tackle those. So I wondered if one or more of you might want to address some of the things that are probably commonly things people hear about in terms of the potential hurdles that we have going forward. Whoever wants to start. Jen?
I'll go first. I guess I'm holding the mic. Well, maybe to piggyback on where I just left, delivery, how you actually deliver the Cas9 protein and the guide RNA into the cell or into the tissue or organ that needs to be edited for therapeutic benefit is still really challenging. There are only a small number of cell types or organ types that are easily addressable today. The blood, and so that's where a lot of folks are working right now, because you can take the blood out of a patient, edit stem cells, edit T cells, edit a variety of other cells that exist in the blood, and re-deliver it back to the patient. And there are really only a couple of other organs, like the liver or the eye, where there are robust delivery technologies. There's a lot of work starting to happen, like a little bit of the work that Jennifer highlighted, to deliver to the brain or to the other organs, but to ultimately be able to meaningfully address even a small fraction of the 7,000 genetic disorders, we're really going to see, we're going to have to see tremendous innovation in terms of new delivery technologies.
Jennifer?
So yeah, I'll just point out that I think what Rachel just said is probably the biggest bottleneck that I see on from a technology perspective for genome editing going forward. And the Innovative Genomics Institute, in partnership with our colleagues here at the Gladstone, are absolutely honing in on this, and we have several projects underway right now that are tackling this challenge with a combination of developing technologies for delivery that already exist, but we're also coming up with some brand new ways that we're very excited about for doing this. And that's really what we, I think, again, taking advantage of this rich environment here in the Bay Area, being innovative about approaches for delivery is going to be key in the future for the success of genome editing in a broad sense. But I also want to point out something else that is very, very important at the Innovative Genomics Institute, and that is thinking about the ethics of genome editing. So there's a number of applications of genome editing that go beyond the kinds of things that you've heard about here so far, meaning making edits to cells in patients and doing that in a way where the changes to the DNA happen in the person, but they don't affect anybody else. But imagine that you could edit the DNA in a human egg or sperm cell or a human embryo. If you do that, then the changes to the DNA become heritable, right? So they can be passed on to future generations. And this is not a pipe dream anymore. This is not a what if anymore. This is actually happening now, right? So labs in various parts of the world are now doing this in human embryos. So far, nobody's used it clinically, so we don't have any CRISPR babies yet. But I think we're all anticipating the day when that happens. And so that's already putting in front of us a challenge. How do we manage a technology that allows us as human beings to control our genetic future? We can actually edit our own species now. It's a profound thing to think about. And so at the Innovative Genomics Institute, from the very beginnings of that organization, we've had a strong interest in helping to lead that conversation. I'm actually part of a group that's been organizing a meeting that will be held next month in Hong Kong to discuss this very topic and encourage an international partnership around this. I'm actually going to the UN. I hadn't had a chance to tell you this yet, but I've just been invited to speak at the UN next spring.
Okay with me.
And about this topic. And I meet pretty regularly with people from governments around the world about this topic, because I think many people appreciate what's coming and what we face in terms of the opportunities, but also the challenge. So I would put that on the table as another big challenge, and it's an opportunity for sure, but it's also a challenge that we face in managing the deployment of this technology going forward.
I should say that Jennifer, this is a real credit to Jennifer. She, unlike many times in science, she saw this early and took the lead rather than having somebody else come and say, wait, what about the ethics? And convened a group of scientists from throughout the world, even it was 3 years ago.
2015.
2015, 3 years ago, to get out in front of this issue and start to think about the ethics. And she's even written a book about this. And many of you know Pitch Johnson, who's one of our legendary venture capitalists in the Bay Area, recently is very interested in this issue also, and has recently given a nice gift to help Jennifer really explore the ethics and social impact of this sort of technology.
Yeah, if there's a question, I'm okay with what you said.
Great. What areas, while you have the microphone, Bruce, what areas outside, Rachel mentioned a lot of people are going for the same disease because it's maybe the lowest hanging fruit, it's tractable. Outside of that, where do you think, and you mentioned the eye, where are the other areas in the body that you think will be attacked first in terms of trying to edit and correct genes in the body?
Yeah, and let me also frame what you just said a bit. There are 4 or 5 major biotech companies that are doing therapeutic editing, and I think all 5 of them either have or will announce programs to edit the same 30 base pairs of DNA, and the reason for that is it really is a good target. It's to actually de-repress fetal hemoglobin, so fetal hemoglobin will come up, but you have essentially all this venture capital and all this tremendous talent, but now focusing on 30 base pairs out of 3 billion base pairs that you could potentially. So that's the condition that we're in, and I think that Rachel started that conversation. But I think that there are, everywhere you look, there are, as we understand the technology, that we see more and more places. It's usually places where we can get to a very discrete area, and we know the genetics very, very well. But my feeling is it's important to take within any of these tissues to take something where you know what the target is, and even if it's only 10 patients that you could treat and then get an effect that is really believable, then you know the delivery problem has been potentially solved. And then once a delivery problem is solved, then you could, 10 more diseases within that potential area will then open up, because now you know it's safe for these 10 people, but now it's ethical to actually proceed to a much larger set. But no venture capitalist and no company is going to finance a program of a disease for which 10 individuals will benefit from, and that's the entire market share. And for very good reasons. But at the same time, it's that kind of work that opens things up. I like to make the analogy to heart surgery, for instance. The first heart surgery was done in 1967. They were happy that their patient lived for a few weeks. The first thing they did when they did the second and third was not how to monetize the next 20. They were thinking about how to do the process better, and I think that that's really the job. It's not the job of the business community necessarily to build the field and to actually to create the basis. That's really the job of the academics, and it's within the academic world that heart surgery went from a very risky procedure to, there have been 3,000 that were done last year. And we can build a field and build this basis for which will I think, potentially affect many, many tissues, many diseases, probably not all 7,000, but because, I mean, some of them involve every cell in your body, but still many diseases. And I can't tell you how appreciative the patients are when you contact them and they've been living with a disease for their entire life and been told that it's completely incurable. And we say, we don't know we have a cure yet at all, but we want to work on it. We want to work with you to potentially try something. Just to have hope is just so gratifying to them and also gratifying to me. I should say that my entire time at Gladstone, or just about my entire time at Gladstone, my entire clinical income has been one medium bottle of red wine, which was given to me by someone I wrote a prescription for. But so it's
about
not
the ethics of that.
Yeah, there you go. All right. But regardless of that, but it's been really fun now to be drawn back by Jennifer. For someone who's coming from the basic sciences, Jennifer really drew me back to see patients again. My practice model, of course, I pay to see them as opposed to vice versa. So,
wow. Thank you, Bruce. That's why we need philanthropy. I do still see patients, and I can tell you the sad truth is that still in 2018, there's so many human diseases for which we don't have a cure, and people are desperate, and we feel that sense of urgency, which is why we're pushing so hard on this effort to really address many of those. I'm going to ask one more question, and then I want to open up to the audience, and this has nothing to do with CRISPR. I have 2 daughters, and I know that for them, it is incredibly inspirational to see women like you see in front of you in the scientific fields achieving the way they have achieved. And I know both of you know that you're role models. Jennifer has been kind enough to inspire my youngest daughter to, she's the biggest Doudna fan ever, as Jennifer knows. But I wonder if you could just maybe both of you comment briefly on what that means to you, what that's been like, and if there's any secret to that.
Sure. Diversity and specifically, I guess in this context, gender diversity is incredibly important to me personally. There are many situations where I'm the only woman in the room. And I think as a result, Caribou is a very different looking organization than your average biotech company. We're about 52 people. Our board is 50-50 women. Our founding team is 50-50. My management team is 50-50, and across the entire organization, we're half men and half women. And I think that's actually a massive competitive advantage for us. We are looking at candidates who other companies overlook, and so that gives us a bigger, better talent pool than other people have. But there's also really clear data, right? If you read a lot of the studies that are coming out of Harvard Business Review and other places, companies with more diverse leadership and more diverse teams win financially. The numbers are really clear. Your return on investment is better. And so I think the answers are obvious, both from an ethical perspective and from a financial performance perspective. And I'd say biotech is in a better place than tech is, but we still have a lot of work to do. The year that Caribou raised our Series A, which was only an $11 million financing, I ended up being one of the top 10 highest fundraising female CEOs in the Bay Area. And I was so upset about that because it was a pretty small Series A, and that was a pretty poor reflection of the gender imbalance. What I'm so excited about is that the pipeline is there. If you look at young women and girls in school, in college, in graduate programs, they're incredibly brilliant people. We need to continue giving them opportunities because they are our future.
So in 2011, we were working away in our lab in Berkeley on this kind of obscure bacterial immune system. And Rachel had been the first brave student in the lab to take on this challenge of studying the molecules that are part of this immune system. And in the course of that work, and really based on her published work that she did as part of her PhD thesis, I was sitting in my office one day and I was thinking, I think some of these molecules are going to be really interesting and really useful. And by the way, this was before Cas9, right? This is before that, right? These were earlier iterations of parts of this immune system that seemed interesting as technologies. And I didn't know anything about business at all, and but I was thinking, there seemed like there was an opportunity. So, I walked into the lab and I said, "Hey, Rachel, I've been thinking that maybe some of these molecules are going to be useful, and maybe we ought to start a company. I don't know. What do you think?" Without even missing a beat, she said, "Of course we should start a company." And she already had, I could see later, she already had a vision in her mind. She already had a map in her, in her head for how we could advance the molecules we were studying in a very fundamental sense in our lab as important tools for biotechnology. And that's really how Caribou got started. So I'm telling you that story because what I've come to see is that, Rachel is a very remarkable person, as you maybe get a little sense of here. But, I think that she and I have, and I often tell her, I could never do her job. I think she's incredible at her job. But I think we have a couple of some things in common. I think we're both people who, we don't let anybody tell us that something can't be done. We're comfortable in our own skin. We do what we think is right from both a scientific and an ethical and a societal and now a business perspective. And we always try to keep those sort of high-level goals in mind, no matter what we're doing, even the smallest minutia. And we both really enjoy team building. She's doing it in the context of a company, and she gets all the credit for building up an incredible team in her company. I enjoy doing it in the context of academic teams and the kinds of collaborations that we're establishing here now at the Gladstone and with Bruce Conklin, of course, one of our early collaborators, and now many others. And so I don't know that those are gender-specific qualities at all, but I think it does speak to the value of just fostering an environment where everyone feels like if they have an idea, it's not about the color of their skin or what gender they are. It's about the quality of their work and their ideas and their values that matter. And that's really the kind of culture that I think we've both really worked to establish, both in our academic and our commercial ventures.
Great, thank you both, and thank you for all you're doing. I'd like to open up to the questions from the audience. I know you guys are always have lots of questions. So, yes.
So, you didn't address one challenge, and I want to know your opinion about it, of that we don't control the repair mechanisms for those cuts that Cas9 makes, and how that makes the results of the editing a little unpredictable sometimes.
Yeah, that's a great point, and I want to hear what my colleagues think about it. But my thought there is, so you pointed out something very important that maybe not everyone understands, or I'll just emphasize it, and that is that the way that CRISPR technology works is it introduces a cut in DNA that then triggers the cell to repair that cut, the cut DNA. And so the cell takes over, the repair machinery is what really does the editing. And I think what you're pointing out, rightfully, is that process, that part of the editing pathway right now is harder to control, right? It's not so trivial. And especially if we want to insert a new piece of DNA, as might be important for some kinds of biomedical and other applications, that's harder to induce cells to do in general. So I'll just say that, first of all, I think that the technology is moving remarkably quickly. And as you probably know, one of the things that makes the technology powerful and in a way is just how accessible it is. So people around the world now have had 6 years to start working on it, figuring out how it works, and then also making it better. So there have been lots of developments that I think are ongoing to control DNA repair and ensure the kinds of outcomes that are going to be beneficial depending on the desired application. And you may know that as part of that, there are now ways of using the CRISPR technology that actually don't involve DNA cutting, right? They involve direct changes to DNA that can be made, or they involve not changing the DNA sequence at all, but changing the output of the genome. And so I think that when we think about the future of this technology, we really have to take all of those kinds of uses into account. I think that the ability to control editing outcomes will continue to advance, but I think in parallel we're seeing increasing opportunities to manipulate genomes in ways that don't involve DNA repair.
Thanks. Yeah, Fred?
Yeah, I just wondering, for the layman here, one, could you briefly describe the distinction between CRISPR editing and recombinant DNA, which we've been hearing about for 20 years?
Sure. So recombinant DNA, which you're right, has been around for quite some time at this point, really just refers to exogenous, an external piece of DNA that is put inside of a cell. And in some cases, the cell will actually pick that up through various mechanisms and stuff it into its genome. That will go randomly somewhere. The scientist usually has no control over where it lands. And often it's used to introduce an entire new gene. So for example, in crop breeding, if you think about Roundup Ready corn, it's using a plant pathogen to actually take a gene from a bacterial species and put it into corn so that the corn plant is resistant to a particular type of pesticide. What gene editing has done is transformed that from a very sort of kludgy, hard or impossible to control process to a much more precise way to take a genome and make changes within that genome. We're typically not talking about taking a piece of DNA or a gene from some other organism and stashing it into this cell. We're talking about making changes within the scope of that genome. Most of the therapies that are under the sort of furthest stages of development so far actually just rely on deleting a gene. To Jennifer's point, deleting is a lot easier right now than precisely inserting. And we're starting to see investigational therapies that are figuring out how to insert a corrective gene using gene editing. And of course, the benefit of that is you get to choose through gene editing exactly where in the cell it lands. The efficiency isn't great, we're working on that, but the precision of the location is there.
Okay, have a question right here? And then here. If you wouldn't mind using the microphone, thank you.
What I was sort of referring to was what we maybe call now traditional gene therapy, it's kind of a cute way to look back at it. But the first attempts at that were replacing genes such as for immunocompromised children or maybe hemophilia. And the first trials were 20 years ago, and we now have 2 approved therapies and maybe 30 people actually treated. So we're sort of hitting that point where it looks like that's going to take off after 2 decades. What's different now? Why, or are we going to see gene editing hit patients faster than that cycle that we saw back then?
So I can take a stab at that. I think that, in fact, that insertional gene therapy, essentially where you take a virus and insert something, has taken a while to ramp up. But what's put into place is that now we actually have the regulatory system, and we have a lot of the knowledge of how to even assess the risk involved, and then we've gotten to approval where it's sort of climbed the mountain. And now, actually, what's happened is that this therapeutic editing is actually a lot less risky in many ways, because we actually can precisely edit in one spot, and we have the tools to actually measure whether or not the edits have happened in places because of the sequencing technology. So when we compare when we compare the risks of making an edit in one gene to, say, take out one disease gene, and then we have the sort of the estimates of what chances of having an off-target effect, and we know exactly where those off-targets are, and we compare that actually to the other therapy, which would be a virus which goes in randomly, and every single insertion is actually a damaging effect, and those the kind of the traditional insertional gene therapy has actually is on its path to approval, it really makes it much easier for us to go in. So I'm very optimistic about benefiting from all the hard work that the traditional insertional gene therapy has had, and then now with this really a technique which has at least the potential in many cases to be safer.
I'd say, Chris, my opinion is long-term gene therapy and gene editing will be complementary approaches. I think for certain diseases, exactly, for certain diseases, gene therapy will be the right choice, and for others, gene editing will be the right choice.
But in fact, the 25 years of development that's occurred in the gene therapy field is actually going to directly facilitate gene editing because of the delivery issue we talked about earlier. The delivery is now going to be enabled by gene therapy, so the Cas9 protein is going to be delivered in a gene therapy manner, and we won't have to redo that for 20 years. That's ready to go. There are several hundred clinical trials going on right now in gene therapy that wasn't true 5-10 years ago. And so I think that's really going to speed up the ability to translate the CRISPR technology. You had a question here?
On the ethical front, is there a current governing body, or what do you envision there? You mentioned the United Nations. I'm just curious what that might look like. It's a sort of science fiction.
Well, yeah, it's an interesting question, because I would say that, you know, at the moment there's a couple things that I see going on. One is that there's a scramble for information. So a lot of people who are outside of the scientific and clinical communities are trying to understand what's going on. They know something's happening, and they know it's kind of big, and they want to know what it is. So that's one thing. And then the other is that there's an effort, I think, very actively among the scientific communities and clinical teams around the world to get together and figure out, you know, how to move forward in a responsible way. And I think that that's very heartening to me. I think the challenge is that I think we all understand that you can't really have regulations that you can enforce globally. How would you do that? It's very, very hard to envision how you would do that. So I think the next best thing is to have, you know, kind of a consensus among clinicians and scientists about appropriate uses of this technology, and perhaps a body like the UN could help to disseminate that. I'm interested to know what I see when I go and speak with the folks there. And I think the scientific societies have a big role to play as well. So I've been heartened by the fact that the national academies in several countries, including in China, have been very much, you know, in the fore in developing the, you know, certainly putting together these conferences and the kinds of reports that are coming out now that advise governments about the uses of this technology. But it's a moving target, and the technology is advancing very, very, very quickly. So it's something that I think we all have to stay on top of as it moves forward.
Thank you. Yes.
Dr. Doudna, thank you so much for your work. My family's been devastated by Huntington's disease, and in Northern California, there's 1,500 people right now sick with HD. It's always fatal, usually strikes young people, and I'm hoping you're going to cure it. It's just a single gene, always dominant. So if the person has it, they'll definitely get Huntington's and die from that, and that killed my husband. But in my family, there's 17 people at risk for Huntington's, and in Northern California, even though there's 1,500 affected, there's 9,000 at risk, and we're desperate. This year celebrates the 25th year that the HD gene was found, and we have not one treatment. Thanks for your efforts. We have big hope in your work.
Well, thank you for sharing your personal story, and I'm really sorry for your loss and your challenge. And I think it's a really good reminder that, you know, it's that we need to stay focused on the goals of all of this in the end, you know. And I think for somebody like me, I'm not a medical doctor, but the idea that someday and hopefully not in the too distant future, you know, the technology that I've been involved in with my colleagues will come to fruition, you know, through the efforts of folks that are now using it to, you know, develop cures, you know, that it will really help patients. I think that's really an incredibly exciting and motivating thing to be thinking about. And I'll just say one other thing about Huntington's, and that is that, I don't know, it's probably almost 6 years ago, that it was right after we had published this work in 2012, describing the CRISPR system as a tool for genome editing. I got a call from a guy at Stanford who said, I'm finishing up my PhD. I want to come to your lab, but I'm not doing anything related to what you do. And I said, well, what are you interested in? He said, well, I'm interested in Huntington's disease. And it turned out that he had been working on the sort of the cellular pathways that go awry in Huntington's, and he wanted to know if he could come to our lab and start developing genome editing to target the Huntington gene, the gene that goes awry in this disease. And so he's actually, his name is Brett Stahl. He's actually been in my lab. He's still in my lab. You know, he came to the lab, and he's led a team. We ended up getting a project funded through Roche, sponsored by a company, to help us advance this technology and deal with the delivery challenge in the brain, which is you know, a big challenge. And so that slide I showed is actually part of his project. And that's been our initial goal, was to really develop a way of delivering this technology that will eventually be useful for Huntington. So we're on it, and we're now starting to partner with people that are going to help us go from where we are today to getting it over the finish line in terms of a real therapy.
And you probably know it's a major focus. Huntington's is at Gladstone, and Steve Finkbeiner, who's here, has been involved in the Huntington's Consortium nationwide to collect families like yours and use human iPS cells to figure out, you know, find new drugs. But you can imagine that's an area where there's an expansion of the DNA, and if you could cut that expansion out, you could potentially address it. So I think there's a lot of hope there. We know it's a devastating disease, but I think there is hope on the horizon. Gary?
As a follow-on to the last questioner, the current administration has approved regulation for right to try for patients with terminal diseases. And does that regulation extend to CRISPR therapeutics? If so, are there trials in progress around that? And if not, is that something that should happen in your opinion?
So, let's see. I'm not familiar with that particular regulation, but I do know that the FDA itself has been relatively proactive recently in trying to look at the regulations involved in gene therapy and how to make them perhaps streamlined. They aren't as streamlined as we would like, and they have actually a draft of the new regulations, and actually through the IGI that we are making comments back on that draft to how to make it even more streamlined. But one thing that they did do was to actually get rid of an entire layer of regulation, which was the RAC, so the recombinant DNA committee. When the gene therapies first came out, the FDA was not actually even capable of taking on these problems. And so they created a committee of specialists, which they called the RAC, that it would have to pass the approval of that before going to the regular FDA. And the current recommendation actually is, because the FDA now has the expertise in the regular parts of the FDA, that they're actually going to be disbanding that committee, which is actually that is a major step forward. But there's other questions which I think are really very interesting, which is the FDA also has a drug side and a device side, and then has a kind of the biologics, which are in the middle. And one of the other things that we're doing also with the IGI and thinking about this is, how can we take not only recommend that they take regulations from the drug side, but also from the device side, because the device side of the FDA has special regulations that are for a device that are made just for 10 people, or just for 100 people, or just for less than 1,000 people. And they have different regulations for those in terms of for safety and for genome editing, and especially the genome surgery type of approach that we have, that the regulations which they can take from the device side could be very valuable. And we're working with device lawyers, and this is another thing that the IGI is very valuable for in terms of bringing in legal advice and other advice to try to think creatively in such a way that we can actually have the regulations be safe. We want them to be safe for the patients. We certainly want them to be safe for the public, so that it goes forward, but to go forward in a way which is as expeditious as possible especially for the patients, like just mentioned.
I think, Gary, your point about the right to try movement here, as well, following on Japan's effort to allow people who don't have anything else to try something, is an important one, and it does open a lot of doors and lowers the barrier to biotech discoveries moving forward. That has had already an effect on the venture community, because the FDA was seen as such a roadblock for so long that people weren't putting money there because it was unpredictable. And I think that climate change is going to be really important for the translation of many of our discoveries in the academic setting to actually get to people. Yes?
My question concerns the societal impact of all this. Well, I think it's wonderful what you're doing, seeing how our society is screaming loud just over modifying plant genes and caring enough to put on yogurt containers where the milk came from, whether the milk came from a cow that was modified in some way or other. How do you think that this is going to affect your work, in fact, even possibly shut you down? Because it's a lot more scary to say you can talk about modifying human genomes than it is whether the corn is herbicide resistant. Sufficiently that you can put Roundup on a field and just not kill the corn, but kill the weeds, which is, I think, the kind you were referring to, and that has caused such screaming and such huge things, and occasionally laws passed and this and that says, oh no, you can't do this here in our jurisdiction or whatever. How do you think that's going to affect what you're actually doing with humans?
Yeah, so through my role at Caribou, I have a fascinating vantage point of getting to work with companies across all these industries and stakeholders across all these industries. And on the food side, there are a lot of questions from consumers. There are a lot of concerns, a lot of which, personally, I don't think are based in science, but not a lot of us use science as a guidepost to what to buy in the grocery store. So I respect the way people make decisions about their food. But that is in stark contrast to virtually every conversation I've ever had on the healthcare side. People bring such hope and optimism to a family member or themselves and their disease, and especially in all of the opportunities that we're talking about here. These are devastating, unmet medical needs where typically these patients have very little time to live or very poor medical outcomes. And I think we have seen across the board tremendous hope and enthusiasm and excitement. So one of my colleagues actually commented years ago, and I thought this was quite funny, there's a decent chance that someday there will be a gene-edited politician arguing against GMO foods. And I love that visual, but it's not crazy. It could happen, right? But I do think there's a tremendous responsibility on those of us in the field to make the best decisions we can, right? You look at the early days of gene therapy, and there were some significant mistakes made, and it set the field back a long time. We learned a tremendous amount from that, but I do think there's a piece of it where everyone's holding their breath a little bit, hoping that everyone is good actors in this field.
No, I can't up
that. Wonderful. Well, maybe one last question. On the front, okay. We'll do two.
Just as a suggestion, don't fall into the same trap that people have done when they've gene-modified plants and everything and called it genetically modified organisms. That would scare even me, and I'm a biochemist and all fully enthusiastic about what you're doing. So find a really good, positive definition of this whole procedure.
New and improved. Is there one more question? Yeah.
Do you feel that the clinical trial framework in the U.S. is prepared to handle like germline editing in the near future?
When I went to the conference that Jennifer organized in 2015, I actually fully expected that there would be a completely new regulatory system made for therapeutic editing. And at that conference were also people from the FDA and people from all over the world. And what was really stunning about that meeting in Washington was that everybody came to the same conclusion, that in fact, when you looked at it, that the regulations that we had in place for the insertional gene therapy, which in many cases, as I pointed out, are actually more dangerous, and also the fact that those therapies, they could go germline, actually. So they could infect gametes and go to the next generation. So that everybody, it was remarkable how everybody came together, and it was not what I thought was going to happen, and not what a lot of people thought was going to happen. Maybe Jennifer knew, but but really there was consensus that we did have the regulations in place in the United States, and that in most other countries. I should say that germline modification of the next generation has already gone on in England, been approved in England, and so we're not actually talking about something in the future. And that was when the mitochondria transplant went in, and the mitochondria went from a healthy person, was transplanted into a person with a mitochondrial disease. That child's already been born. That child, when they have a child, will carry that mitochondria. So germline modification in humans has already occurred and has actually happened under complete guidance of what goes on in England, and I think that it's actually being done in the United States now. The big step really in this whole germline question was in vitro fertilization. When in vitro fertilization first happened, that was once babies start being born with that, then there was acceptance by that. But that's actually was the biggest step. Once that actually happened, then the rest is actually relatively small changes in my mind. CRISPR is a major thing. Obviously, I'm a big fan of CRISPR, but actually, once you actually have in vitro fertilization, then you can do PGD, which is essentially a pre-implantation genetic testing. Once you do that, you're already selecting the next generation. And that's actually for 98% of genetic diseases, PGD is actually all that would be necessary, and without any CRISPR, and that's already selecting a bit of the next generation. So essentially, we're already partway there in reality with just in vitro fertilization, with just sequencing, and then also it's already happened with mitochondrial transfer. So it's not, and I'm not any more afraid of what's going on now than before. So I have confidence that we have the regulations in place. I also have fears, though, of course, that it will be abused and all that other things, but I don't necessarily think that more regulations are necessary, because the more regulations wouldn't actually change what happens in some part of Inner Mongolia or something like that. We can't control every part of the globe.
Well, I hope what you've gathered today and what you can feel is the incredible excitement and where we sit in history on finally being able to understand and address some of the most devastating diseases that you've heard some about already today. There's really a lot to be hopeful for. It won't happen by chance. It's going to happen by very purposeful, committed, long-term effort, and that is what we're here to do. But I've never been more excited than I am today about what we can do for human suffering. I want to close with borrowing a phrase that I heard last night. It was an event with Tom Steyer. His wife, Kat Taylor, is here. And we were talking about climate change and hunger. And Tom framed those as matters of life and death, which they are. What we're talking about is really a matter of life and death. And so we have to work the hardest we can with the greatest sense of urgency and commitment, and that's what I assure you that we will do here. So let me just close and thank Will Evers and Fred Dorey, who lead our President's Council, for making this event possible, our speakers, and we do have some more wine and gene-modified hors d'oeuvres out there. Thank you. Thank you.